Welcome to another video of irradiation processing of foods. In this section, we are going to delve deep into the mechanism of irradiation. Now, let us step back and look at where the idea came from. So the basic methodology for producing shelf-stable irradiated foods, particularly meat, seafood, and poultry products, was developed over several decades by the United States Army. This was roughly around the 1950s to the end of the 1970s. And that makes sense, does it not? So in the past, probably even to some extent today, military operations often require foods that are lightweight, shelf-stable, microbiologically safe, and ready to use under extreme conditions. You cannot rely on refrigeration on every setting, and you certainly do not want foodborne illnesses in the field. So military food systems have often pushed innovation in preservation technologies, including irradiation. Now here's something very interesting about these products. They were normally packed without gravy or a sauce or brine. This is quite different from canned meat products, which we are familiar with, where the liquid often plays an important role in heat transfer during the sterilization process. So, liquid in canned food products plays an important role because it facilitates what we know as heat transfer during this sterilization process. So on the other hand, in canning, the liquid helps to distribute the heat through the product, but with radiation, this is not necessary. So what you have here is the ionizing radiation. It penetrates the packaged product directly, so you do not need to surround the meat with liquid just to achieve sterility. And so what this does is that it gives a product with different sensory profile. Instead of a casserole-like flavor that many people associate with canned meats, these irradiated products, they tend to have a more dried cooked taste, and more like a roast meat. So even from a quality and consumer perception point of view, what happens is that the product is not simply a copy of the canned product or canned meat, it's a different category altogether. And so this is an important reminder that food preservation for us does not just influence shelf life and safety, but they also shape the flavor, the texture, the convenience, the packaging design, and ultimately the consumer experience. Now, let's make this real. Food irradiation is not just a concept that is buried in textbooks or hidden in old research papers. It has been used commercially, and right here in South Africa, we've got shelf-stable irradiated meat products that have been produced on a commercial basis by the Biogram Group at the Atomic Energy Corporation of South Africa. Now think about the challenge that we are trying to solve here. How do you produce a meat-like product that is safe, stable, and usable in remote environments, in rural areas, where refrigeration may not be reliable or it may not exist at all? So the answer was not simply to cook the meat and hope for the best. The process was much more controlled. So these meats were first cooked, then chilled, portioned in vacuum packed, and finally irradiated. So what the irradiation does is that they use the irradiation dose used was around 4 to 5 kilogray, and this was done at a very low temperatures between -20 and -40 degrees Celsius. The question is, why irradiate at such low temperatures? And if you think about it, it's because low temperature helps to protect the product quality while still allowing the radiation to achieve the required level of sterility. In other words, the product becomes microbiologically stable without being ruined in the process. So that is the balancing act in food processing: safety on one side and then quality on the other. So the packaging here, ladies and gentlemen, also matters because the products were packaged in high quality 4-layer laminate pouches designed to withstand tough conditions. Unlike metal cans, these pouches would not rust, and they were less likely to burst under harsh environmental conditions. As long as the package remained intact, the product could be guaranteed for more than 2 years. That is a very strong shelf life claim, especially for a meat product. And so the question is, who used these products? Not ordinary supermarket shoppers popping in for a quick braai pack. These products were specifically valuable for people who are doing safari operators in remote areas of Africa, mountaineers, yachtsmen, canoeists, and genealogical surveys teams. So people who are also doing camping and hiking for a long period of time. So in other words, these products are designed for people operating far from cold chains, supermarkets, and convenience. So this is a very practical example of how food science respond to real logistical problems. Now we move into the science. When ionizing radiation passes through food, it interacts at the molecular level. And so two key processes occur here. First is ionization, where the electrons are removed from atoms, and there's a free radical formation, which is the second process, where highly reactive species are created. These free radicals, then they damage the microbial DNA, they disrupt the enzymes, and then they alter the cellular structures. Think of it like this. You are not burning the microorganisms, you are disabling its biological machinery. So when we ask the question, what is food irradiation, we are simply expecting a response that says food irradiation and microwave heating, they employ radiant energies that produce their effects when absorbed by the food. So both of these technologies, they require specialized equipment for the generation of energy and the protection of human beings. So there are several forms of radiation energy from different sources. And this belongs to what we call the electromagnetic spectrum. So we've got various kinds of sources. As you can see in this diagram, we've got the infrared light with 800 nanometers or longer to raise the temperature. And then we've got energy radiations that are limited from the background of atomic structure. Such materials are said to be radioactive. Then we've got materials such as uranium, which are radioactive, and cobalt-60 that can make radioactive compounds by bombardment. So if you look at the electromagnetic spectrum, the cell falls around the infrared spectrum, and then bacteria towards 10-6, towards the viruses, which are in the ultraviolet region, and then proteins, soft rays, and then you've got water molecules under hard rays, right? So as you can see, if you go over this side, which is to my right, these are shorter wavelengths, and then if you go to my left towards the houses, these are longer wavelengths. So here towards the 10 to the power 3, we are operating at the radio waves. And then if you go down here, we've got the cell going down here to the shorter wavelengths. The cell is bigger than the bacteria is bigger than the virus, and the virus is bigger than the protein, and the protein is bigger than the water molecules. So here you've got shorter wavelengths. So as I've said, the infrared light, it's around 800 nanometers or longer, and raises the temperature. There are several forms of radiant energy, and these belong to the electromagnetic spectrum, as I've said. And then we also said there are two ways in which radiation affects the food systems. Firstly, you've got what we call the direct effects, where radiation hits the critical molecule directly, and you also have the indirect effects, where you've got radiation that interacts with water, forming free radicals. Now let us consider this. Most food products, or most foods, are high in water. So in reality, the indirect effects, they are going to dominate. So let's make an example. In fresh meat, radiation produces hydroxy radicals from water, and these radicals, they attack the microbial cells. This is why cooling moisture and oxygen is important in this process. So now let's delve deep into these kinds and sources of energy. I want us to start with something that is familiar, UV light. I think you've all seen UV light being used in water treatment, in surface sterilization, even in some laboratory cabinets. Now in food systems, UV light is in the range of 200 to 280 nanometers, and this is particularly effective at achieving surface inactivation of microorganisms. But here is the catch, and this is important for us to note. UV light has very poor penetrating properties. So what does that mean practically? It means UV will only work on the surface. For example, if you expose a piece of meat to UV, only the outer layer is treated. Anything that is inside, it remains unaffected. That is why UV is widely used for equipment surfaces, conveyor belts, packaging materials. It's not used for bulk food preservation. Now, if you compare that with X-rays, X-rays have much greater penetrating power. They can move deeper into the food systems, but they come with a limitation. And they are very difficult to control and focus efficiently, especially at commercial scale. So most applications remain experimental, particularly with X-rays, rather than being widely used industrially. Now we shift to the third one, which is what really matters in food irradiation. And so in food irradiation, we are going to be talking about ionizing radiation. So this is where we actually use what we actually use in the food industry, and it's often referred as cold sterilization. So why cold? It's because unlike heat, it does not significantly raise the temperature of the food, and this is key for preserving quality. So these have got a greater penetrating power and have the inability to produce significant radioactivity in food materials. So now let us look at the spectrum of radiant energy. Not all radiation is equal in food processing. At one extreme, we've got radio waves similar to what I've shown in the previous picture. These have very long wavelengths and they have no germicidal effect. Then if we move to infrared, this generates heat, so its effect is thermal rather than microbial inactivation. And then if we talk about visible light, what you and I see every day, that is very little, it has very little antimicrobial effect. So where do things start to change? Things begin to change when we enter the ultraviolet region. Here, especially at the 200 to 280 nanometers, we get maximum germicidal activity. I think this is where the DNA absorption occurs, leading to microbial inactivation. But still, we have the same limitation. Penetration is low. Now we then need to go even further into shorter wavelengths. This is where we reach ionizing radiation. So ionizing radiation is what we use in food application, and this is what we are going to center our discussion around. Let us now move into the core of food irradiation, ionizing radiation and its sources. When we talk about ionizing radiation, we are referring to the energy that is released when radioactive isotopes decay. So these isotopes can occur naturally or they can be produced artificially in nuclear reactors. As they decay, they release different types of particles and radiation, each with very different properties. For the purposes of this content, you are required to understand 4 types for this chapter. First, we're going to talk about alpha, we're going to talk about beta, gamma, and then we're going to talk about the neutrons. What do we understand about alpha? These are essentially helium atoms that have at least 2 electrons. They are relatively heavy and carry a positive charge. But there's an important part that we need to understand. They have very low penetrating power. They cannot even pass through a sheet of paper. And then secondly, the beta. These are high energy electrons. They are lighter and more penetrating than the alpha particles, but they are limited. A thin sheet of aluminum is enough to stop them. And then the third one is gamma. These are not particles, but they are energy photons. They are extremely penetrating. They can pass through dense materials, which is exactly why they are useful in food processing. And finally, you've got the neutrons. These are highly energetic particles that can interact with atomic nuclei. Now, here is where you need to be very, very, very careful. Not all of these radiations are suitable for food, because each type has a different penetrating power and, more importantly, a different interaction with matter. Alpha is too weak, beta is moderate, gamma is highly penetrating, and neutrons are extremely energetic and can alter atomic structures. So already you should be asking yourself which of these we should actually use in food systems and which ones we should avoid. Keep that question in mind, because we are going to try to answer that in the next slide. Now let's answer this question directly. Which radiations are suitable for food applications? The answer is very clear: gamma rays and beta particles. These are what we call food compatible emissions. Why is this the case? Because they have sufficient penetrating power to inactivate microorganisms and enzymes within the food without causing the food to become radioactive. This distinction, ladies and gentlemen, is very critical. Now, if you contrast that with the neutrons they carry very high energy, and when they interact with atoms in food, they can actually damage the atomic structure. So what happens is that this is going to lead to induced radioactivity, and that is completely unacceptable in food processing. So neutrons are not used. Now let us talk about where these useful radiations come from. Gamma and beta radiations are used in food processing are typically derived from spent nuclear fuel, which is carefully controlled and shielded. The radiation source is never in direct contact with the food. Instead, the food is passed through the radiation field in a controlled system. So you would have heard us talking about cobalt-60. So this is one of the most widely used sources in the industry. Cobalt-60 is important and special because it emits gamma radiation, which is highly penetrating and very effective at microbial inactivation. So if you think back to what we've discussed earlier on about penetration, gamma radiation allows us to treat food through its entire volume, not just the surface. This is what it makes it so powerful compared to something like a UV light. So I want you to take some few moments and answer these two questions for yourselves.